Maayong adlaw mga katusok, this is Faijah Marohomsalic of BSMLS 4C and welcome to MedTech for the Day. In this video, I will be showing you different laboratory tests, procedures, and techniques that are done under the microbiology section of the laboratory. But before anything else, disinfection is a must. First is plate streaking. This is done to obtain an isolated colonies from inoculum. First is to prepare the materials needed and apply antiseptic technique to the inoculating loop and dish every streaking. Second, get the isolated colony that you wish to isolate. For the first streaking, create a straight vertical line and streak it horizontally. Rotate the dish 90 degrees for the second streaking and pass through the first streaking twice. Lastly, rotate the dish 90 degrees for the last streaking and pass through the previous once only. Now the streaking is done, apply antiseptic technique with the loop and turn off the alcohol lamp. Now that the procedure is done, always make sure to disinfect our workspace and dispose all the waste. Second is gram staining. It is done in the microbiology section to check for bacteria at the site of suspected infection. To start, disinfection should be done to avoid contamination. Also, labeling should be done before conducting the test. It should include the patient's name, date, and type of test. It is important in the laboratory to label the specimen, materials, and even reagent to avoid errors and cross-contamination. And also, it will help the medical technologies to release a reliable and accurate results. The first step in gram staining is to prepare the smear. Apply antiseptic technique with the slide by passing it through to the alcohol lamp 2 to 3 times both sides. Next is to apply antiseptic technique with the loop and dish to get the isolate. Put the inoculate in the slide and spread it in a circular motion until you achieve a very thin layer. After the inoculation, apply antiseptic technique with the loop. Next is heat fixing by passing through the opposite side of the slide 2 to 3 times through the flame. And now we can proceed with the gram staining. First is applying the crystal violet for 1 minute. Next is to wash it with distilled water for 5 seconds and make sure not to heat the smear directly. Second is the grams iodine and apply it for 1 minute. After 1 minute, wash it with distilled water. Third is the decolorizer. Apply it for less than 30 seconds. Then wash it with distilled water. And the last stain for gram staining is saffron in red. Make sure that you apply it for less than 10 seconds and immediately wash it with distilled water. After staining, let it air dry. Now that the slide is ready, you can view it under the microscope using the oil immersion objective. With this, now you can identify whether there is a bacteria present. Third is catalase test. Catalase test facilitates the detection of the enzyme catalase in the bacteria. Also, it is particularly important test to determine whether the gram-positive cocci is a streptococci or a staphylococci. First is to prepare the materials that you will be using during the procedure. Next is to label the slide with the patient's name, date, and type of test. Second is to apply antiseptic technique with the slide. To inoculate, apply antiseptic technique with the loop. And get an isolate. After getting the isolate, spread it in the labeled slide. Before putting the loop down, apply antiseptic technique. Next is to add a drop of hydrogen peroxide to the slide and observe for the effervescence. Presence of effervescence is interpreted as positive. Fourth test is coagulase test, which identifies whether an organism produces the exoenzyme coagulase. To perform this, apply antiseptic technique to the tube to get the plasma. Next is to add the human plasma in the labeled tube. To inoculate, apply antiseptic technique with the loop and get the isolate. Next is to add the inoculate into the human plasma and apply antiseptic technique. To acquire a result, tube should be incubated. And lastly is biochemical tests. These tests are among the most important methods for microbial identification. Some of these tests are the TSI, LIA, citrate, urease, and SIM. First test is TSI, and the inoculation is stub and strict. Second test is LIA, and the inoculation is stub, strict, and stub. And the third test is citrate, and the inoculation is strict. And lastly is SIM, and the inoculation is stub. Always remember to apply antiseptic technique before and after inoculation. Every after procedure, dispose the slides properly and the waste in the proper bin. And of course, after handling different specimens and conducting different tests, hand washing is a must. This is to maintain cleanliness and safety. This has been Faijah Marohomsalic, your medtech for today. Thank you for watching.